So when I was at the FDA for about 15 years, this is kind of before, the genomic revolution. Really, this was in the 90s, right? We're just developing some of the underpinning platforms and sequencing the genome. Still inside the FDA, it dawned on a number of people that, while this genomics revolution was taking place at the regulatory level and even at the commercialization level, many of the analytes, many of the diagnostic technologies, and in some instances, many of the therapeutics were protein-based. And so, the field of proteomics, which is extraordinarily nascent, in the 90s, still, is lagging technologically behind than the genomic side, I think was appreciated and in some ways has as a fundamental philosophy kind of rooted in that, almost all of the FDA approved targeted therapies in oncology, their mechanism of action is against proteins and protein activity. Indeed, with even markers like PSA, CRP, CA19-9, CA125, those are protein-based markers. And the FDA is really used to kind of regulating that process and working with the diagnostics industry. In the proteomic field, actually, it was or pre-genome, which is interesting philosophically, and now that's certainly not the case now with many of the, BRAF V600E and every, NTRK fusions and things that are coming in are part of a package insert. That's changing, and so it's really a multi-omics kind of view right now to the agency. But I wanted to start that off as a preamble because this is something that I've been interested in on the regulatory side and now on the academic side of how do we effectively incorporate new types of proteomic technologies, maybe protein information, and translate that to kind of bedside applications. Just for disclosures, I have financial relationships to disclose. I'm a consultant for today's talk. The RPPA technology has been licensed to Avant Diagnostics. I'm consulting for them. I have patents that have been licensed out so that you guys know that. I always like to disclose everything. So look, as I said, the DNA is the hardware of the cell. It's the proteins and metabolites and other translators of the biochemistry or the software of the cell. Most often the drug target itself, sometimes the therapy. In the Center for Biologics, I was responsible for oversight for things like interferon alpha and interferon beta, which themselves are protein-based therapies. So, these are some screenshots that are actually pretty ancient now, but these were coming out right at the beginning of, the TCGA projects where they were, the kind of the Atlas type projects, what were the genomes of cancer at the very nascent level. And of course, at the time, right, one of the big aha moments was at the atomic level, at the molecular level, every patient's tumor seemed to be different. And so it was like, wow, the heterogeneity, and I'm not even talking about clonal heterogeneity, right? That's a whole topic of seminar for itself, but just take a tumor, sequence it, and then sequence different patients that all seem to be histologically identical in the microscope. And at the, the single nucleotide level, different. And so, of course, now it's like, what are the drivers, the passengers? What are the things we should target? What's actionable? That's the whole rigmarole now. But it was like, well, how is this going to work? Is every trial an N of one trial? I mean, how do we translate this heterogeneity into, subgroups of response, et cetera? And so one of the things that, bioinformatics efforts do is they try to coalesce this heterogeneity into, functional pathways. Well, these are, PI3 kinase pathway kind of mutation family, or these are, ERK family, mutation path. And so we try to categorize these into maybe target pathways that are shared. Well, these pathways really functionally are protein and enzymatic and metabolitic pathways at a functional level. So we're really wanting to understand not infer. So it's like, let's not infer pathway activation or pathway engagement by RNA or DNA. Let's layer onto that very important information, because that's the foundational information. Are the pathways actually activated or not? And we can develop technologies that might be able to measure these, and we'll see how robust those technologies are. But we can grow with the tools that are available to us. So we're interested in this, and I think, again, with my former FDA hat, if you look at the package insert of many of these targeted agents, right, and I say, before you go to bed at night, read the package insert, and it'll put you right to sleep, right? So, but within the package insert, there's a lot of information, obviously. And when I was at the FDA, we actually spent quite a bit of time with the industry partners, wording that package insert is actually part of the approval process, not just the drug. A lot of people don't realize that package insert is actually an approved process. So if you look at, say, Afinitor, right, it's widely used for ER-positive breast cancer patients and kidney cancer and a variety of other tumors. You look at section 12 under clinical pharmacology, it's always there, mechanism of action. It gives you some clues to what the drug company and the agency feels are important information for patients. I'm just giving you just one as an example, right? Everolimus, inhibitor of mTOR, it's a kinase. It inhibits the kinase activity, the activity of S6 protein kinase and 4EBP1, downstream effectors. You don't see anything about a gene mutation here or transcript level. We're talking about enzymatic downstream members. And so, trying to develop technologies that might be able to actually measure these things would be important. And then you could look to see, well, is measuring them actually important, right? So as John and I were talking, develop technologies, assess them rigorously, and then you can maybe ask, well, so what? Okay, is it useful? Maybe it's not. Maybe just knowing it's ER positive is enough for the use, right? That's the approved label use is ER positive patients. As we know, these cartoons that are in every one of these papers, review papers, or even in our catalogs from the antibody companies, they draw these, even if they're complicated, these linear pathways. We know biochemically that that's not the case, right? It's much more of a rat's nest of feedback loops and protein complex interactions that we're just starting to understand. So that anybody that comes in, and if you talk to folks like Chuck Sawyers or people that have been involved in, say, just like one pathway, they'll be like, we're still just trying to figure out what that communicative network really is. We have to be cognizant of, when our technology development side, that we're playing with a card deck that maybe we know like 3 of the 52 cards, right? And we're still scratching the surface at these interaction networks, but we're trying to develop technologies that can measure as much of this content as efficiently as possible. And I would say from a proteomic level right now, we're kind of, what's the cognate from the genomic side would be, well, what's the targeted exome panel that we really, if we're going to focus on like 400 things and not 40,000 things, what are the ones that we need? And we may need to measure the 40,000 things first, right? And so there's that background of global proteomic analysis, targeted proteomic analysis. Same, I think, similarity philosophically as, whole genomic sequencing, targeted exome. How much do we really need? What's the bang for the buck? And how do you interpret that data and translate it, is huge. And so, we have understanding of some of the basic precepts, especially the proteomic side, of signal transduction and how kinases, which are obviously very important as drug targets in oncology, transduce a signal through SH2, SH3 interactions. We know how modifications like phosphorylation become a very important mediator of that process. And of course, there's other modifications that are also very important. But we do have some fundamental understanding of how protein signaling works, and that gives us a hook into understanding some of the functional activity of this. So, when I was in graduate school, understand protein phosphorylation, we might do like a gel shift assay or nuclear run-on assay. And, the lab was screaming with gamma-labeled phosphorylated, ATP. And, so you were having to do a lot of incorporation assays. And this was before kind of phosphospecific antibodies came around, or the ability to measure more directly with reagents that are more translatable to clinical diagnostics. And so I wanted to spend a little bit of time before I got into what we're doing with a little bit of hey, this isn't just our idea. People have been very much interested in looking at how do we layer in things like measuring protein phosphorylation and its relative importance to understanding prognostics, prediction to specific therapies. And this is a series of papers where, when they would look at, gene and mutation analysis versus protein and phosphoprotein analysis in similar studies, what do we see? Well, in this paper, right, they looked at the role of EGFR, RAS, PIK3CA in predicting EGFR TCA efficiency in patients with advanced stage squamous cell non-small cell carcinoma. They did not observe, and they looked at some observational studies, right? So that in this specific type of cancer, not necessarily predicted by mutation status alone. So unlike maybe adenocarcinoma, where there's truly, exon 20, T790M, alterations can, show resistance and even response. Well, and then Dan Hayes, right, when you would go into, bread and butter, response to erlotinib and gefitinib, known FDA-approved, on-label EGFR inhibitors in the intended use population, you would be able to predict response not just to the therapy by the EGFR mutation status, but looking at phosphorylation of the substrate of the drug target itself and downstream substrates like AKT. And you could do this regardless of EGFR mutation status, right? Because we knew in these early studies, yes, while patients with EGFR mutation status certainly were enriched for response, there were EGFR wild type patients that responded quite nicely to erlotinib and gefitinib. And of course, the converse is true, right? Responders not, or mutation carriers not responding to the therapy itself right away. If they had some type of de novo resistance, right? Or that may not have been a driver of that tumor, even though it was a genomic alteration. And, groups went back and looked at retrospective study sets where they had a response, where the drugs were given in unselected populations. They were able to go back and say, all right, erlotinib given to EGFR wild type, let's go back and look at what's correlating with response now that we have some new reagents available to us that maybe weren't available to us when the trial initiated. So these folks went back and they used phosphospecific antibodies to the EGFR receptor, so tyrosine 1068, and they found that in an EGFR wild type background, they could discriminate responders and non-responders simply by EGFR phosphorylation, even in a wild type setting. Now, this Kaplan-Meier curve, the responding population isn't quite as good as an EGFR mutation-based population, but it's pretty close. And so the interesting thing here was going back. Now the horse is out of the barn, right? So, the drug companies are not going to change their companion diagnostic regime to incorporate this in. This is prospective analysis of a retrospectively, collected set, but it sets the basis of there's kind of gold in them there hills, right? There's some signal there that's missed by simply going in and doing a genomics analysis. And we have a lot more of these, right? So, when they've gone in and looked at response to ER-targeted therapy and looked at PIK3 mutation analysis in these patients, in these breast cancer patients, this was in these Stockholm studies, where they're able to go in and look at large cohorts of thousands of patients given some endocrine therapy, for identification of companion diagnostics, the focus should switch from analysis of to the analysis of activated proteins in these pathways, which are associated with adjuvant tamoxifen resistance. And specific protein targets emerged and looked to be potentially dichotomizable. where you can look at low AKT-mTOR activation versus high AKT-mTOR activation by phosphorylated IHC-based work, and you could show that in these patients, again, prospective analysis of retrospective set where you know outcomes, and you were able to say, well, geez, when this pathway was not on, you certainly got benefit. But when the pathway was high and ostensibly on, and mTOR is a known kind of a chemoresistance pathway, you lost the long-term benefit of tamoxifen in this group. So people are interested. So a lot of it was, well, how do we go in and kind of explore the signaling architecture if we're getting some hints that maybe measuring, this specific phosphoprotein can maybe add value or identify patients like the EGFR patients, those patients that were phosphopositive but wild type would never get put on therapy right now, right? That's not an indicated population. In a tumor board discussion, they would be put on other types depending on other drivers that may have arisen from their NGS analysis. But potentially could be responsive if they were EGFR phosphopositive. And that's not a routine CLIA test that's being done. So how do you translate the information kind of discovery process to maybe routine clinical use? Start to get into that. Ultimately, philosophically, what we're trying to do is develop technologies that, if really understanding the signaling network is an important part of the landscape of a systems biology landscape of what you'd like to see at the tumor, what do we need to do to be able to measure, these pathways at a functional level? Well, we have barriers to entry technologically that we have to consider. And these are just some, right? You probably, if we come up here with a chalkboard, you probably could extend this list many more. But these are some of the big ones that I think, kind of rise to the top. Look, tissue is the issue. You have very small amounts of material in these precision medicine studies, right? And often limited to FNAs or EUS obtained biopsies or core needle biopsies, very small amounts of cells, limited amount of tissue. And, I talked to my colleagues like Forrest White, fantastic, earth-shattering papers in Cell, comprehensive phosphorylation analysis, massive global profiling, huge bioinformatics, starting amounts in the trillions of cells, right? So what you can do in cell culture, not even close to what you would be able to do in clinical material, which also speaks for the discovery side of proteomics and then kind of the clinical proteomics side. And you really winnow down the technologies because every technology has their pros and cons. And so discovery proteomics, difficult to translate into, a few hundred cells in a single slide. And you're used to working with milligram quantities of input, and you're talking about picogram quantities and lower. And then, tissue preservation and fixation at tertiary sites is limited to formal. We're not going to change the pathology workflow anytime soon. We've actually been funded by the NIH to come up with alternative fixatives, like that are molecularly friendly. Extremely difficult to drive those into pathology, even if it's positioned as a precision medicine fixative. And the point is, right, it's in the US especially, almost all cancer, especially metastatic, are treated at the community level. They have one way that they treat those cancers it's drop it into formalin, it's routine pathology, and that's a train that's left the station, right? Only if it was part of a package insert where you forced, from a payment standpoint, the pathologist is not going to get paid unless they do it this way, or you're going to change that process. So we have to be able to transition from maybe discovery in frozen to does it work in formalin? And that's a lot of things falling off the cliff there too. And that's sometimes a little bit frustrating, right? Because it's like you may be a biochemical truth, but you won't be able to translate it into a commercial setting because your analyte of interest, is gone, degraded, cross-linked, the CVs start to spread out. You can't discriminate anymore. And frozen, it was rock solid. And so you go through this process of due diligence as you translate this. Tissue heterogeneity issues are huge, and clonal heterogeneity, tissue heterogeneity even at the macroscopic level. And really the force of the oncologist community now In the US, they're used to, I'm sending this to Foundation Medicine, I'm getting my 367, it's multiplex. They don't even use the the actual like Opus FDA approved test for say measuring ALK rearrangement. So I'm just going to I'll measure ALK on Foundation Medicine and 366 other things, right? So their appetite for multiplex testing is there, and they expect some similar things from the proteomics field that would drift into the clinical setting. And we're seeing this pressure, multiplexed IHC, CyTOF-based panels, MRM-based panels. People are we're a panel-focused clinical world right now, perhaps for good reason, in the context of trying to figure out how best to treat a progressing metastatic patient. And so that has to be both quantitative and multiplexed. And then even more so in these cooperative studies, right, where we have small amounts of material, it's you have to feed a lot of little birds in the nest, right, with with a single piece of tissue. And I can tell you that, genomic and IHC based they're top feeders. They get all the tissue. And then your exploratory new thing, gets the end of the end, and oftentimes we'll have no more tissue left, right? Or the study was 300 and there's 50 patients that have some tissue left and you're left with small n. So this is some practical issues right now that's facing the clinical proteomics community of even if you have some interesting technologies, how do you kind of, shove your way in line and get access so that you can kind of prove your merit, prove your worth. So this background in some ways underpinned a lot of the motivation for us to develop a technology called the reverse phase protein array to try to address some of these, I wouldn't say I would say address in a collaborative way because it's just one type of technology that can be implemented. But we developed this with an eye towards some of these technical barriers. And that was almost a nano dot blot like technology that could use extremely small amounts of input material and could deliver quantitative and multiplexed information along the way. Now, we did not care about the cost. We did not care about the turnaround time when we developed this. It was a technological, can we quantitatively measure the signaling architecture? And could we develop a robust method and transition this in our lab into a CLIA environment and a CAP environment? And we were fortunate when we developed this at the NIH, where we had this collaboration between the FDA and the NCI, and I was able to work with Lance Leota's group, who basically was doing all the AP and CP for the NIH. And so they had a CLIA lab, 100 pathologists, lots of residents. And so we were able to kind of develop this in a CLIA CAP environment, even from the very beginning, even in an RUO side. So this technology does away with the need for, say, sandwich type assays because it's a dot blot type assay where you're immobilizing this onto a economic substrate like nitrocellulose that has high binding capacity and could actually bind denatured proteins. Because one of the things we realized early on was a lot of these phospho-specific antibodies and modification-specific antibodies really required a linearization of the epitope to give you kind of robust analytical results, kind of technical results, reproducibility, precision, accuracy, intra-assay, intra-operator type stuff that you have to start thinking about when you're developing a CLIA test and developing the reagents to be able to assess that robustness. But it requires, just like immunohistochemistry, really well-validated primary antibodies, right? You're hanging your entire hat. And the array doesn't have the advantage of having spatial orientation on it that allows you to do some due diligence by IHC on the performance. You see nuclear staining, and it's like you see the punctate, you lose all that. So you really have to go in with well-validated antibodies because it's a single site, and then you're amplifying that primary antibody. Now we use CLIA cap type based reagents, in fact, FDA approved type reagents on the back end for the amplification. So we adopted the Hercept test, the biotinyltyramide amplification system. We actually broke the kit and used that back end material for the readout. And we used the DACO or Ventana autostainers to do it. So you could use things that are part of like the IHC process to use in an array-based process. So we were kind of thinking early on, like, well, how could this perhaps at one day maybe transition over? And we were still thinking pretty rudimentary, and still are in many ways. But it adapts to clinical workflows that pathologists are more used to. And so this array is printed, and we can print 1,000 patients on one array and technical triplicate. We can basically print about a nanoliter of material of lysate. So imagine you making a lysate that you'd run on a Western blot and taking that same lysate from that well and transferring it to like 500 arrays and doing 500 things with that, lysate that has only a few micrograms of protein in it. And you can get the multiplexing by basically printing many, many arrays. This also is nice because it allows you to potentially develop calibrated assays because everything on that array is seeing the same primary antibody, secondary antibody, all the downstream analyte detecting and amplifying reagents, everything's seeing. So you can do a lot of real robust analysis because you're looking at everything within the same array. So the hope, and we'll get into that, the hope is that you can go through a workflow. From a tumor biopsy, and I'll show you why, we go through microdissection for all of our analysis. But the point would be to kind of start panel-based analysis by drug target mechanism of action and looking at, can we measure the signaling network around EGFR signaling if you're looking at an EGFR inhibitor, or AKT signaling if you're looking at an AKT inhibitor, and looking just at a lot of signaling pathways to see if there's coordinate feedback networks, or you find markers that you may not have thought predict response that are or off-target effects. So we've gone through in our lab kind of an idea technology to tech transfer. We've trained a large number of laboratories around the United States that operate in kind of a core facility for research-based applications. We've kind of taken a different path because developed the technology, got it out there, published lots of papers. We're really into now trying to get this commercialized and, more importantly, analyzing its impact in a CLIA kind of cap-based setting. And we're one of the few proteomics labs in the United States on the academic side where we have a cap-accredited CLIA licensed laboratory. So we're able to transition our research findings over into a formal environment and then test that in clinical trials where we actually have, and I'll show you examples, where the information from the array goes to tumor board for patient decision making. So it's not just research, it's being evaluated in that setting. So we're really kind of pushing that side of things. The ability to do so leads to a lot of self-evident, I think, attributes that are great for clinical implementation. So low abundance, way below limits of sensitivity that are accessible by mass spectrometry, even by triple quad MS. Requires only one antibody, and you can do quantitative analysis, really vastly exceeding kind of Western blotting and immunohistochemistry. Now, the question would be, do you need that or not? So we have a whole series of calibrators and controls, high and low controls we developed for dozens, not hundreds, but dozens of important analytes. So we've gone through and well-validated antibodies in a whole validation series under CLIA, develop calibrators that span the linear population range, high and low controls that can be transitioned right above and below defined cut points that you print on the same arrays. We have some of these that we've printed on arrays we've run weekly for 6 years. And we have that kind of trend data across about 30 analytes that we're really interested in to see what the inter and intra-operator lot number of antibody changeovers, we can monitor those things, which is very important. We have a library of antibodies that we continuously validate. We do not develop antibodies in our group. We buy them commercially and vet them. And as many of us know, what the company says Like this is what it looks like on Western. We run it. Sometimes it's like doesn't look anything like what the company says. So we kind of are trust but verify type of thing. Well, not say the companies are hogwash, but it's like let's see what it's like in our hands with our lysates, with our high and low controls. We'll do a lot of peptide competition analysis and phosphatase deployment of, and doing forced expression type work where we know clean signal. So I would say about 20% validate, right? About 80% do not validate in our hands, which definitely constrains from a CLIA standpoint what we really kind of believe and will bring forward. So a lot of work needs to be done on that end for sure. But at the end of the day, this technology allows us to measure hundreds of drug targets and downstream signaling proteins at once quantitatively from microscopic amounts of material, from laser capture microdissected epithelium or stroma or endothelial cells. We can detect about 100 molecules per spot, and it depends obviously on the affinity and avidity of the antibody, but it's very, very sensitive assay in that regard. And now it's being used by lots of laboratories to look at lots of different things, right? Because of its multiplex nature and ability to look robustly. So, we use this to model the NCI 60 library. We did, very much a systems biology view across the phosphoprotein architecture all the way through 60 other different omics, including drug response prediction and confluency analysis. Gavin Macbeth's group has done a lot of work at looking at, tyrosine kinase network signaling modeling. There's a lot of, it's a group, it's a technology now that has its own society, that people come together to share resources about what antibodies are working, not working, new methods of detection. So it's a robust technology in that right, and lots of people are using it. So these are just some snapshots, right, of the technologies used in different types of, it's now an integrated part of TCGA and some of the larger moonshot projects. However, as any technology is, it's only as good as what goes into it, right? So it's the GIGO kind of, garbage in, garbage out. So one of the things we're very focused on is, okay, the technology seems to work really well analytically, technically. However, what's the impact of some of the upfront processes part? Cancer is a product of the tissue microenvironment. As you all know, here's just a home out prostate specimen. You have every one of these cell types and more. And unlike the genome, right, the proteome is a constant fluctuating information archive. Every cell type has kind of its own proteome, if you think about it kind of functionally. And even in the context of those cell types within an individual's microenvironment, the proteome is nothing more than, in some ways, a functional snapshot of what the cells are doing biochemically at that point in time. Oh, and by the way, that also might be reflective of how the tissue has been taken out and sitting on the bench before it's even put into formalin, and how long does formalin take to penetrate? About 1 millimeter an hour. So you have all these, and the cell's still alive, and the proteome is not happy. It's not the in vivo state that it's used to. So you have to be very careful because you're measuring a little bit of a canary in a coal mine with the signaling architecture, especially similar to RNA expression analysis, where you're getting the cell's reaction not only to the underpinning biology, what's happening at the DNA level, but also just what the cells are seeing. So we've been very interested in incorporating upfront technologies like laser dissection or cellular enrichment technologies upfront, and look at the impact of that. And we have new automated LCM technologies. There's different types of laser dissection, ablation, direct capture, automation, no automation. So they're out there. And we've gone into looking at just some fundamental things like, okay, does subjecting the cells to laser energy change the phosphoproteome, right? Just some very standard in the grass type of, so we went through, where we took, we microdissected the tumor epithelium from the cases, and then we just took the whole specimen, we just laser dissected non-specifically, just subjected the whole field, and then we lysed, and these were contiguous sections that we numbered 1, 2, 3, And then we 1, 2, 3, 1, 2, 3, 1, 2, 3, and we pulled all the ones, pulled all the twos, threes. All the ones were microdissected. All the twos were just kind of subjecting the whole tissue to the laser, and 3 was don't do anything at all. Across a couple different study sets, we said, okay, unsupervised clustering, heterogeneity aside, you know, do we see any real effects of the laser on the cells? And we did not. We've done about 6 published studies where we've looked across different tumor types and even non-cancer types just to see what the impact of the laser is. And then we've looked at study sets where we've actually had some really interesting. So we trained Gordon Mills' group at MD Anderson, who's did a lot of the TCGA, RPPA data. We had groups of people. We had a group of bioinformatics people from Memorial Sloan Kettering contact our lab thinking that we actually generated the RPPA data, and they were complaining about some discordances between the RPPA protein data and the genomic data in some of these studies. And we're like, we didn't do it, but what exactly are you talking about? And they were like, well, one of the things is EGFR and PTEN protein and phosphoprotein levels are not correlating with like PTEN LOH mutation or EGFR mutation status. We know for these patients they should, and we're not seeing it. We're like, all right, well, we didn't do it, but one of the things that the MD Anderson group didn't do was microdissect. They just took the tissue, ground it up, and analyzed it. So maybe maybe that's the reason, because the RPPA technologically we kind of stand behind, and Gordon's group does a really good job in the RPPA. So it's like, So we actually went back and they gave us some of the sets from a GBM study set that they use for TCGA and said, all right, let's take some of your 39 TCGA glioblastomas and let's look at a subset of proteins and phosphoproteins, including the PTEN and EGFR pathway that you say were not correlating. And we bread loafed it. We took alternating sections and we either laser dissected or not those, and then we pooled. So we got no cellular bias in any one. It was like about 40 sections, and we just went, and we took 20, microdissected, 20 did not, pooled everything, and then we ran them all in the same array. And what we found, sure enough, is that only the microdissected glial cells had the required correlations with the downstream genomics. So looking at P10, the LCM in the top panel, looking at P10, irrespective of the underpinning tumor contents, because some people are like, well, if there's a high enough amount of tumor, maybe it doesn't matter. So we looked at like greater than 90% tumor. And still, the SL is slide lysate, which is basically just lysing the cell. Only in the microdissected did you see correlation between mutation status and phosphorylation of P10 with P10 copy number. You'll see only the LCM had the necessary expected expression differences and phosphoprotein. When you didn't, you lost it all. If you just look, and this is what Memorial saw. It's like, we don't see any correlation. It's like, yeah, only because it was non-microdissected. This is actually the TCGA data itself. So we went in, and this is just, we stained for P10, and you see endothelial cells staining for P10, another cellular structure staining for P10. And the point is that at the protein, it's very important what the input is, because unlike looking at, say, a rare alteration or an alteration like, say, BRAF V600E, and the concern is, do I have enough tumor there to be able to see that low noise in the field of, say, normal BRAF background DNA? The proteome, every cell may be producing some expression of that protein or phosphoprotein. It's just a relative, and you don't know ahead of time, because that's the point. You're trying to find out what that relative expression differences are. So just grinding up this tumor and asking for P10, you'll get a perhaps very technically precise answer, but it will be inaccurate. So it's always the distinction between precision and accuracy. And in clinical medicine, accuracy trumps precision, because that's are you getting the true answer? You want it to be both accurate and precise, believe me, but accuracy always trumps precision in that aspect. So here is an example of perhaps something that was highly precise but not accurate. So when we go into our RPPA technology, we go through a microdissection process, printing, staining, image analysis, extraction. And now for the remainder of the talk, I'm going to give you some vignettes and how we're using this at the bedside. So initially in the I-SPY trial, which is a neoadjuvant breast cancer trial that our lab is involved with. This is the first trial, I-SPY 1. It was just exploratory. I-SPY 1 was just a standard neoadjuvant trial, patients coming in and getting basically ACT therapy during the course of their neoadjuvant regime. But we were able to get access to core biopsies taken at different points in time, and we had complete pathologic response to the backbone chemotherapy that would eventually be used in I-SPY 2. So it was a nice little sandbox for us. And we did, RPP LCM RPP analysis, and we saw, distinct, there's a heat map, red are high levels of activation, green are low levels, and black are kind of the intermediate levels like you'd see by gene expression. But these are, a cadre of phospho and total protein on this side. So you can see the kind of these unsupervised kind of clustering, and we can look for structure, and we did, but certainly heterogeneity, right, at this signaling level, patients with high levels of HER2 activation or MAP kinase pathway activation, et cetera. And what do I mean by activation? Well, when we look at some of the subgroups formed, and so this is just a snapshot of the triple negative tumors from I-SPY 1, we see, pathway architecture emerging, right? So self-organized kind of, we see EGFR activation and downstream, kind of IRS activation and SHIC, adapter proteins. They're going up and down together, if you will, even though it's a snapshot in time. AKT with two of its kinase substrates. So just we see structure, and we were able to go in and do comparisons with kind of rock solid FDA approved ways of measuring proteins like IHC and FISH for HER2. And this is the RPPA data. So you can see this is just the quantitative relative amounts of HER2, and then in green is HER2 positive by IHC, 3+, HER2 negative by IHC, and then a couple that were indeterminate. You can see on the continuum, largely nice correlation, but the explosive nature of the quantitative aspects of measuring these at a cellular level, you can get huge fold differences in the relative amounts of the protein in a dichotomized population. This is HER2. So with no false positives, pretty good concordance. Same, and this is FISH. So here's the HER2 to CEP17 ratio, total HER2. You largely see the HER2 negatives, all low, and the HER2 positives higher, and a couple HER2 positives that are low, which is interesting. Two of those three were negative by IHC, so some of the disconcordances between IHC and FISH. But again, these are all kind of HER2 positive no matter which way you slice it, by the array, by FISH. And these are all negative, and you had some, like I said, disconcordances. With a general cut point of no false positives that, are pretty good, worth, 3 in some ways false positive, but good concordance. But when we looked at the phosphorylation level, we saw something interesting. So if you look at this population of patients that are all HER2 negative by FISH, by RPPA, HER2, and now you look at phosphorylation level, there's a subgroup of these patients, now we're looking at phospho-HER2, that are FISH negative, HER2 negative, but phospho-HER2 positive. Very interesting, because their levels of phosphorylation of HER2 were the same as the HER2 positive group, but again, by phospho are all positive as well. At a practical standpoint, thinking about it, this is looking at the activation of the protein. None of these patients will ever get a HER2 targeted therapy. All of these patients were, but just looking at whether or not the protein is activated, those patients have similar looking like pathway activation architecture. And it's not seen by any other assay. So it's not an intrinsic subtype component. It's not a PAM50 subtype. It's not HER2 expression found. These are not they're not HER2 mutated. So it was like, something's going on in this group that has this pathway activated. Now, this is observational. And it seemed that this group also had downstream signaling intact, like phosphorylation of EGFR was also higher in this group. And some of the downstream components. We looked, is this not just HER2 high, but did you see heterodimerization potentially occurring and downstream signaling occurring? And the answer is yes. In this subgroup that were FISH negative, IHC negative, but phospho-positive, there also seems to be this kind of structure. It's about 10%, significant minority, we found in this cohort seemed to be pathway activated. And of course, Soon-Peg published in his study from, I think this is the B31 study, about 10% of the patients that were found to be central HER2 negative benefited from trastuzumab. So that's kind of interesting, completely observational and kind of inferential. So we went back, now that we were now fast forward into I-SPY 2, where we have patients that are coming through a trial where they're getting randomized to different types of therapies. In this trial, they graduate and they move along. So they have eligibility criteria in I-SPY 2 that's largely driven by a MammaPrint scoring, and the only patient population that's not eligible are the HR positive, HER2 negatives that are MammaPrint low. Everybody else comes in, and they graduate if they reach a statistical significance in a Bayesian analysis that actually looks at significance of potential graduation in a phase 3 setting. Don Barry's group has kind of pioneered this kind of cut point that's not path CR based, but more whether or not the drug would graduate in a phase 3 setting, which you'd have to run, but it's a probabilistic determination. So what we had in this study is a number of drugs that are coming through that provide a sandbox for us to look at the RPPA type signaling architecture to see if we get correlative analysis. Neratinib, which is an EGFR HER2 kinase inhibitor, was given to HER2 negative patients. So it allowed us to test the hypothesis, right, that we saw in I-SPY 1 of this group that was phospho HER2 positive. Now we actually have patients that are given a HER2 targeted drug, because we could say, so what? Is that population important in identifying? So yes, finally we have an inhibitor that against at least the HER2 part in this study. So in that study population, the drug, neratinib, graduated in the HER2 positive HR negative population. It did not graduate in other subtype, although you can see the triple negatives wasn't without some hint of sniff of a signal. Certainly compared to HR positives, you saw some enrichment in the true triple negative group. And so, and Jill Gray had some cell line work that seemed to indicate that triple negatives may be more sensitive to neratinib, and they thought it was on the EGFR side. So, neratinib hits kinase inhibitor. It's not an antibody, right? So it's on the intracellular side. So based upon that mechanism of action, we postulated in a prescribed manner, in a qualitative manner, meaning a pre-prescribed, that HER2 phosphorylation, EGFR phosphorylation, in the pre-treatment sample would associate with response. We measured about 300 phosphoproteins. There was full RNA, transcript analysis, DNA analysis, intrinsic subtype analysis. The only markers to predict response were phosphorylation of HER2 and EGFR, the pretreatment biopsy sample. So we hit a home run. It was a prescriptive, qualitative biomarker. It was a hypothesis we generated ahead of time. We actually did not define a cut point because we still had to explore in this cohort, so it wasn't a grand slam. But we said we were going to predict that this associates with response, and it did, and it survived all the different interaction analysis. Yep. So we published a paper that showed that measuring the phosphorylation of these two proteins in a triple negative population was able to identify a subset of patients that otherwise would be missed entirely by all other molecular profiling that you could do. And it was a significant population, and we found actually that in triple negative patients, this drug may be especially active because a subset of these patients, this is a scatter plot of HER2 activation and EGFR activation. So this is the control arm and the treatment arm. You can see all the responders are in the EGFR phospho and HER2 phospho high. So this is a HER2 EGFR dimerization event, and the drug hits both. And so this may be perfectly tuned to a population of patients that if you enriched for both phospho EGFR and HER2 in an ostensibly HER2 negative population and a triple negative population, actually achieves a very good pathologic response. So if you look at the double positives, we have a 77% path CR rate compared to what the drug graduated in. And so we're very interested in this because the prevalence is about 43% of triple negatives appear to have this. So we're now transitioning this to a CLIA setting prospectively now with PUMA. It's not again, this is not a HER2 mutation or genomically driven event because the prevalence is so low. So I'll stop there. We're at a point now where we're going to be transitioning this finding. This is just a vignette, lots of vignettes, that we have an observation in a study set. We're going in an independent study set in FFPE because this was all frozen. We want to see if this holds up in FFPE samples where we'd establish a cut point, and then PUMA would use that cut point to select patients in a phase 3 setting if we see the same thing occurring. And we'd be doing this now solely under a design control in our CLIA labs. So with that, be happy to take any questions.